stands for Lysogeny broth, and it's the most common type of bacterial media, so basically food that we feed and grow bacteria in. And it's the most commonly used, but that doesn't mean it's the best. People just kind of use it because it works. But other things might work better for various purposes. So, for example, if you're doing a protein expression, you're trying to get these bacteria to make protein for you, you might want to look at TB. TB is terrific broth. It's going to be richer. It's going to have more nutrients, more amino acids and things that the bacteria need in order to make proteins. You're going to be able to grow to a higher density, so get more bacteria in the same volume. So TB can be good for that. If you're doing something with, you want the DNA to be copying, or you want the bacteria to be copying DNA, like a plasmid, you might want to use something like SOB or SOC. These have magnesium sulfate, which is going to help the DNA copying molecules kind of grab onto the DNA. SOC is going to also have glucose, which makes it really good for recovery after transformation. And so I'm going to go over all of this, as well as some other more specialized types of media and things like this that you might encounter in the lab. But yeah, so LB is the main one, but not the only one by far, and probably not the best one for what you're doing, but it works for most things, so yeah.
Biochemists and molecular biologists, we get bacteria to do a lot of work for us. Everything from copying DNA, such as from plasmids, to making proteins, expression. And I have much more on those in other posts, but today I wanted to talk about how we pay the bacteria because they're not going to work for free. And how we pay them is by feeding them. And so there are a lot of different bacteria foods that we use, and we call these foods media. And there are different types of bacterial media that are good for different purposes. And I want to go over some of the main ones, as well as some of the various definitions and terms we might use to describe media, such as defined media, where all of the components are every ingredient is known exactly how much of each ingredient there is, as well as undefined media, like your classic media, such as LB, which has things like tryptone, which is basically chopped up proteins, and you don't know exactly what's in there. But it has everything that the bacteria will need. You're just not exactly sure what it is that you're giving it. And so it'll differ from batch to batch. And we'll get into all of this. The main media you're probably going to come across is LB. LB stands for Lysogeny Broth. You might sometimes see it's like attributed as standing for Luria Broth, but it actually stands for Lysogeny Broth. And it gets this name from Giuseppe Bertani, who first published this recipe in 1951 when he was using it to study lysogeny. And lysogeny is a step in the sort of infection cycle of a type of bacterial infecting virus, so bacteriophage, or commonly is shortened as phage. Basically, some of these phages, what they do is they can enter this lysogeny state where they infect the bacteria and they kind of hide out in the bacteria. They insert their DNA into the bacteria DNA and kind of bide their time until the conditions are right to enter a lytic phase where they break out. So he was using this media to study this lysogeny phase, and so he called it Lysogeny broth. And so later others would make variations on this LB. And so one of the people who made some variations was Luria. And so it's often referred to as Luria broth, but Luria was kind of just building off of Giuseppe's recipe, but lowering the salt concentration. So what's in LB? One of the main things in LB is tryptone. And so tryptone is basically the chopped up protein, chopped up casein protein. And it's a type of peptones. So peptones is basically a word we use for a mix of protein pieces that we get from chopping up proteins. And in the case of tryptone, the chopped up protein that we're chopping up is casein. And so this is like one of the main milk proteins and that sort of thing. And so this gets chopped up by pancreatic enzymes to give you this tryptone. And this is a good source of amino acids, so of protein building blocks, so that we can get the cells to make lots of protein that we need them to make, whether it's for their own stuff or for proteins we want them to make for us. But that's not all that, so this is going to be your source of amino acids, but that's not all the bacteria are going to need. some of the stuff that they're going to need, well, we're not exactly sure. And so what we do is we add yeast extract. And so this is just going to be like broken open yeast. And so whatever was in the yeast is good enough for the yeast. Maybe it's good enough for the bacteria. So this is going to provide a source of all the vitamins and trace elements, minerals, and things like this that the bacteria might need. And again, tryptone and yeast extract are going to be examples of kind of like undefined components. You're going to get batch to batch variability depending on how the digestion went, how much of various protein there was, what was in that yeast extract, things like this. Also, what we know that they're going to want is sodium chloride. And they don't need it just for themselves, but they also need it so that we can kind of keep their environment happy in terms of the osmotic balance. So how much water there is inside and outside of the cells. If you were to place bacteria in really dilute media without a lot of salt in it, what would happen is that you would have this, we would say that the environment would be hypoosmotic. Basically, there's a lot more stuff inside of the cell than outside of the cell. And so water is going to start flowing in from outside of the cell into the cell, and this is going to cause the cells to swell up and burst. And so by adding table salt outside, we're increasing the amount of stuff out there. And this is going to help make it so that the water is not flowing in and making the cells swell up. It's also going to help with transport and other things. And different recipes are going to vary in how much of the salt that there is. So Miller and Bertani, they have a high salt. Lennox is also going to be higher, and then Luria is going to be lower than your Lysogeny broth. Now, Bertani's recipe, he also included glucose, so just your good old like blood sugar. And so, well, it's just a source of energy and things like this, but not all the recipes are going to use glucose. So LB is going to be your main media, and we'll talk about some other alternatives for different purposes. But first, I want to just mention that we can use this media in various forms. So we can basically have the liquid media that we just grow bacteria in suspension. And we can do this at different scales. We can do just like in a little tube, or we can do in a big flask. Or what we can do is we can actually stick this media into an agar gel if we want to grow bacteria on like a petri dish. And this is really good if we want to isolate individual colonies. So we can get bacteria to kind of take root and then grow on top of each other. And this is going to give you these like genetically distinct colonies where the bacteria, because they're multiplying on top of each other, each of the bacteria in the colony should be like identical. Whereas if you had them growing in suspension, they'd all kind of be mixing together. And so we can grow bacteria on these petri dishes. And in these petri dishes, you basically have a gel mesh that's made up of this agarose sugar, this agar sugar, and it's going to be filled with this growth media. And sometimes we use selection media, where basically we take those plates and we stick an antibiotic or some other selection thing in there. Basically, if we stick a plasmid with the genetic instructions for getting the bacteria to do something we want to make sure that we only feed the bacteria that actually took in that plasmid. And so in addition to having the genetic information we want the bacteria to use to make a protein, say, we would also have a selection marker such as an antibiotic resistance gene. And this way, only the cells that actually take in the plasmid will be able to survive in the presence of that antibiotic. And so that is an example of a selectable selection media. And often, and we can again do the selection in the plates or in the liquid media, depending on our purposes. Before I move on to these plates, though, I just want to say a cool fact. Apparently, the first gelling agent tried to make those bacterial, those plates, I like to think of them as kind of like bacterial pancakes. The first gelling agent tried was actually gelatin. Robert Koch tried it in 1881. Basically, the bacteria were like, ooh, gelatin, let's eat this. And that also was not very good because we want them, to be growing in the liquid part of the media and not just like eating the mesh, eating the gel matrix. And also that gel melted too easily. But the wife of one of Koch's associates, Angelina Fanny Elshmus, suggested that he try agar. And so he did, and it worked really well. The bacteria didn't eat it, so you still have that nice gel mesh with the media running throughout it. The bacteria can live in and on, and it stayed solid. And it didn't kill the bacteria, which is always an important thing. So that is where we get the agar plates from. But now let's talk about some of the other media that we might use. There are a few different options depending on what you're trying to do. If we're trying to express a protein, one of the media that we often try is Terrific Broth. So this is abbreviated TB. It's richer than the LB, and by richer, basically it has more of the yeast extract and more of the tryptone. So it's going to have more of the amino acid, more of the vitamins and minerals. And so this can make it so that the cells can grow to higher densities and so that they can actually make more protein. So you might get better protein yield. And so often you might say, try expressing a protein in both TB and LB and seeing which works better. It also has pH buffers. So basically one of the reasons why bacteria can't grow to high density in just LB is because the pH of the media is going to get all messed up. But by adding this potassium phosphate components, you get this buffering system where it's going to keep the pH more stable. And this is going to prevent the cells from dying, even though they're growing at high concentrations. And so TB is going to be a richer media that's going to be good for protein expression and growing at high densities. Another media with more of the tryptone and yeast is going to be your 2XYT. It gets this name because it has twice the yeast extract and the tryptone. So it's good for long growth times because there's enough nutrients to last you a while. But it differs from the Terrific Broth, both in it has a little less of the yeast and the tryptone, as well as it doesn't have the buffering agent. Two other common ones you might see are SOB and SOC. SOB stands for Super Optimal Broth. This one, it has more excess tryptone, and it also has magnesium sulfate and potassium chloride. So magnesium sulfate, basically this is good for molecules that need to work with DNA, such as DNA polymerase, so the DNA copying machinery. So DNA polymerase relies on using magnesium to help it interact with DNA molecules to hold onto them, as well as to get DNA molecules to link together. Basically, DNA has a negatively charged backbone thanks to its negatively charged phosphate groups, and magnesium is positively charged, so it's going to kind of help neutralize the charge of the DNA backbone so that these DNA pieces don't kind of repel one another and that you can actually stick them together. So it relies on this magnesium sulfate, and this SOB has additional magnesium sulfate in order to help promote these sort of processes. So SOB as well as SOC, they both have magnesium sulfate. So these are going to be good for when you're trying to do DNA copying, such as when you want these bacteria to be making lots of copies of a plasmid for you. SOC, this stands for Super Optimal Broth with Catabolite Repression. And this is also going to have glucose. So it's going to have the magnesium sulfates, just like the Super Optimal Broth. And it's going to have the extra tryptone, just like the Super Optimal Broth. But it's also going to have glucose. And this is going to make it good for post-transformation recovery. So just like you might eat one of those like sugar gels or something if you were to run a marathon, to help you recover, we can give these cells glucose in order to help them recover by giving them a source of energy that they don't have to work very hard for. And so often we're using this to help them recover after like a transformation. So basically in transformation, we can stick a plasmid into bacteria to give them the genetic instructions for doing what we want them to do. But this can involve kind of making them unhappy. We have to chemically weaken these cells or somehow electroporate them or something to basically open up pores in their membrane and let this plasmid in. It's kind of harsh on these bacteria, and so we need to let them recover afterwards. And the SOC can help them recover because it has that glucose. In terms of the name, so SOC, stands for Super Optimal Broth with catabolite repression. So catabolism is basically the breakdown of molecules. And so a catabolite is a molecule that is formed as like an intermediary or an end product of a breakdown process. When bacteria break down some energy sources, then you get the buildup of various catabolites that can actually inhibit the bacteria from growing more, for copying more. It can change what type of food the bacteria start using. And so by adding extra glucose, you're going to help prevent the buildup of those products that occur when the glucose is running low. And this is going to help promote the bacteria to keep doing things as normal and being very active. And so super optimal broth with catabolite repression can help with that. When we're on the process of catabolites, basically some of the breakdown products, such as of lactose, can actually get the bacteria to switch their food source. So initially they're growing on glucose and they like growing on glucose, but if they run out of glucose, then they can switch to like using lactose. And we can take advantage of this in order to kind of trick them by adding a fake catabolite, IPTG, in order to trick them into using this like lac promoter and things in order to do inducible expression. So to get them to think, okay, it's time now we switch to using to making those genes that we need to make lactose or to use lactose, but instead of having instead of them actually starting to make those genes, we trick them into making a protein that we want because we've stuck the promoter, the regulatory region that's normally in front of those lactose making genes, we stick it in front of the gene for getting them to do what we want. And now these cells will do what we want. There's also this really cool media that I want to try where instead of just like adding IPTG for the induction, it's actually an auto induction media. And so basically it has a defined amount of glucose and lactose. And so basically the bacteria use the glucose until they run out, and then they switch to using the lactose. And things are calculated so that the bacteria at the time they'll grow lots and lots. And then at the time when they run out of glucose, it's the perfect time for them to switch over to lactose. And so it's like this auto induction media. So I've actually like made the media and stuff, and I really want to try it out. But I haven't had the opportunity yet. But maybe in the future, I'll do a post on that. Those are all examples of undefined media. So they're going to have that batch to batch variability, thanks mainly to the tryptone and the yeast extract, which we're not exactly sure what's in it because it's going to depend on what was in that protein mix, how those peptones were digested, or how those proteins were digested, how much, what the yeast had eaten, all these various things can then influence what's in the food, and this can influence the batch to batch variability, which can influence the experimental results. And so sometimes this isn't a big deal, especially if you're just getting them to make protein or something. You're not trying to study the bacteria themselves, you're just trying to use the bacteria. So it could influence your yield and stuff like this, but it's probably not going to have a big impact on your experimental results. And so often this is going to be fine for us. But say you want to actually study the bacteria, now you're going to probably need a more defined media. So a defined media is going to have a constant components. And so an example of this is M9 minimal media. It contains essential salts and nitrogen. So it contains sodium phosphate, potassium phosphate, ammonium chloride, and sodium chloride. Where the heck does the name M9 come from? I am sorry to say that I still do not know. So you might think, oh, it has 9 components. No, it's got 4 components. Okay, so where does it come from? I got really excited because I found this reference to this Miller book, and I actually found it when I was at Cold Spring Harbor for my PhD, and I found a copy of it in my library, and I went and I looked it up, and basically they just have the recipe for the M9 media, and then they use the M9 media, but they don't tell you where the heck the name M9 comes from. So it's probably just like the 9th thing they tried or something like this. Sometimes with defined media, you actually add extra ingredients. So minimal media, basically you just want to give the bacteria enough that they need to survive, but you might need to add extra things. And so some of these extra things you might need to add depend on what you're trying to do and what type of bacteria you're trying to use. Something that the bacteria can make themselves, we say that they're autotrophic for. And something that the bacteria can't make themselves, but they need, we say that they're oxytrophic. So basically, imagine you had these bacteria that they need coffee to survive. So it's essential for them. If you give it to them, if you had bacteria that can make the coffee themselves and bacteria that can't make the coffee themselves, if you were to grow both of them in media with coffee, you wouldn't be able to tell them apart. But if you grow them in media without coffee, not only the bacteria that can make the coffee, the bacteria that are autotrophic for it can survive. And so this is going to allow you to select for the bacteria that actually have the ability to make the coffee. We can use various variations in this oxytrophic, autotrophic strategy in order to do different things and test things. A common place that this comes into play is with the Ames test, which is a test for mutagenicity. So basically whether a chemical compound can cause mutations. You take bacteria that are oxytrophic for histidine, for making histidine, which is one of the amino acids, and you grow them in media that doesn't have histidine. So these bacteria have a mutation that makes it so that they can't grow unless you give them histidine. But then you treat these bacteria with a chemical, and that chemical, if it can cause mutations, there's lots and lots and lots of bacteria, so there's lots and lots and lots of chances to have a random mutation that's going to convert them from oxytrophic, so from needing to be given back the histidine, to autotrophic, so being able to make it themselves. So you're going to like fix that original mutation. You get this reverted mutant. And if you get this reversion mutation, then these bacteria can grow even if you don't give them histidine. And so in this way, we're able to see, okay, well, how many of the bacteria were able to acquire a mutation that fixed them. And so the more bacteria that acquire this mutation, the more mutagenic the compound likely is. So much more on that in other posts. That is an example of selection. So basically with selection media, we're only allowing things to grow if they have a certain trait. We also have screening media. So with screening, we're basically saying, okay, you can let everything grow, but show me, somehow differentiate between the ones that have a trait and those that don't. So make them look a different color, say. And this is the idea with blue-white screening, where basically bacteria, if they have an uninterrupted gene for making this protein, they'll be able to turn this colorless compound blue, and if they don't have that gene, or that gene is interrupted, then it will be white. And so in this way, the bacteria will look different colors whether or not they have that intact gene. And you can use this to do things like look and see whether your cloning might have worked. In any of these cases, you're going to want to sterilize the media, and so we typically use an autoclave, which is basically like this really hot, kind of like sauna. It gets really hot and high pressure, and it's going to sterilize that media. And then we can use that media in various forms, various flasks and shakers, and things like this. So to review, bacteria food is called media. We can use it in liquid and in gel matrices. The most common one you're probably going to come across is Lysogeny broth, so LB. This is the most common. It's basically good for a lot of different things. It's not necessarily the best, and if you talk to a microbiologist, they might tell you it's a pretty cruddy media in general, but it does the trick for most things, and it's nice and simple, easy to make. You can often buy just like a mixture that's pre-made, then you just mix it in water and sterilize it, or you can make it up from the different components and note that they smell. It's kind of like an acquired smell, whether or not you like it, but the tryptone, especially when you're weighing it out, can be a bit of a whiff. But anyway, Lysogeny broth is going to be that main broth, and it's going to be kind of like a lightish gold. You might also see some darker broths. You have your TB and your SOB and your SOC. Basically, the TB is going to be a richer media. It's going to have more yeast and tryptone, so more nutrients. It's going to have potassium phosphate buffers. So this is all going to help the bacteria grow to higher concentrations without dying. This can be good if you want to get them to make a lot of protein for you in a smaller volume. Note that when you're using if you're using Terrific Broth, You're going to, if you're doing an IPTG induction, you're going to induce at higher ODs. Basically, what's going to happen is because you need the cells can grow to a higher density, you want to let them grow to a higher density before you induce them. So before you tell them to start making protein. In terms of SOB and SOC, these both have magnesium sulfate, which is going to make them good for DNA copying. So for making copies of plasmids, because that magnesium sulfate can be used by DNA polymerase and other molecules that work with DNA. SOC also has glucose, which is going to make it good for post-transformation recovery. So when you're basically allowing the cells to recover after a transformation, so after you use heat shock or something to get a plasmid inside them, and you want to let them recover before you like add antibiotics and plate them and everything like this, the glucose can help them with that recovery step. And then you can switch them to growing onto LB, such as in your Petri dish. These are all examples of undefined media because you have batch to batch variation. There's also defined media, but that's going to be more tedious to make, more expensive, and this sort of thing. And these basic media often do the trick for our basic biochemistry needs. But if you want to study things about like what's going on in the bacteria themselves, you're probably going to have to be a little more careful with the composition of the media. And you can manipulate that composition in order to select for things and screen for things and do all sorts of different cool stuff.